Um, so welcome everybody to this forum where it's a pleasure for me to introduce Professor Uma Ramakrishnan from the National Center for Biological Sciences in Bangalore, in India. Professor Ramakrishnan got her undergraduate degree in Bangalore and then a PhD at UC San Diego, after which she was postdoc at Stanford, and since then, almost 20 years, I think, has been a professor at NCBS, where she has focused on aspects of ecology, population genetics, studying both humans and animals, using molecular methods to understand the larger scale questions that ecology has asked for millennia, but now really made powerful precisely because of the ability to essentially get molecular tools to essentially play a role. I think it would be better if Professor Ramakrishnan tells us what she is working on rather than me describe much more. I just want to point out one thing. She just got elected to the American Academy of Arts and Sciences, and that's why she's here. So congratulations. Over to you to tell us something about the genetics and the future of India's biodiversity.
Thank you. Thanks everyone for coming, and it's really nice to be here. I kind of invited myself, so I just thought it would be fun to share not the details. This is not a technical talk at all. It's more of a kind of a bigger picture, kind of a synthetic, almost how I think, or musings in a way of how I've been wandering through my career. So as Maha said, I'm interested in, my training is in population genetics, and I'm very interested in molecular ecology. And through my career, I've been very keen on trying to think about biodiversity and conservation. And actually, when I was doing my PhD, it was almost considered to be a bad thing to be interested in conservation science. And it's only the last 10 years, maybe 15 years, that we are really realizing that the challenges that we face are to a large extent environmental. And this is very true, I think, especially in the context of where I'm from, India, where I think the challenges that India as a nation will face and Indians will face are a lot about the environment. And yet our understanding of many of these things remains poor. So yeah, I have the controls here. So yeah, so basically, India is an amazing place. We have, amazing ecosystems. This is from the Western Ghats, grasslands. There's a Barasinga deer in Kanha. We have the high mountains in the Himalayas. The elevation, the average elevation in the Tibetan plateau is 4,000 meters, right? It's a crazy habitat. And we also have spectacular species. Here you can see two tigers kind of battling it out in Tadoba Tiger Reserve. And of course, large herbivores like elephants. Here they are in Bandipur National Park, where they're walking on the road. There's a herd, and you can see there's a baby over there, and there's a car right behind kind of following them. But of course, habitats in India are very, very fragmented. This is a drone picture, again, from the Western Ghats, showing how it's really, it's kind of, I think it's difficult to imagine if you have not been there, but there's really, humans and wildlife all right intermixed with each other. It's not like the Amazon, where you have a huge hulking amount of area, and there's very good segregation maybe. This is also how it looks. So for example, if you look at, these are basically the past reconstructions of forest cover and human settlements and so on. So if you look at India in the 1700s compared to 2000, you can see that there's been, forest cover has gone from 60% to 9% forest cover, or green cover, let's say. And villages and agriculture have increased from 20% to almost 78% land area. You can also immediately see that basically is in very, very small patches. So what really remains is, very, very, very fragmented. And yet, I think it's amazing that in this, most populous country now in the world, we are now the most populous nation in the world with a growing economy and only 5% of land area protected. This is against For example, the global recommendation of 15% of land area to be protected. We still have a lot of biodiversity. We still have these amazing ecosystems, and even more, I think, basic, Indians depend on biodiversity. So people in India depend on biodiversity for livelihoods, whether it is, forest products like honey and, harvest of all kinds of things like mahua and all kinds of things. There's a lot of dependence of people on biodiversity for livelihoods. For services, all of our rivers originate in the Western Ghats and the Himalayas. In fact, the Himalayan glaciers basically provide water to a third of the world, right, between India and China. And also, very, very importantly, something which we're realizing more and more, these remaining patches of biodiversity do mitigate climate effects. So there's been a lot of recent work showing hydrology, for example, that forests allow for more catchment of rainfall and so on and so forth. And these are things which are slowly disappearing as these habitats disappear. So of course, it's critical to sustain and conserve biodiversity in India, but it's clearly a very complex system, and that's how hopefully I was able to communicate to you. So I told you that I'm a population geneticist, and you must be wondering, what am I talking about? Livelihoods and all that. I don't work on in the social sciences, but I think what I'll do today is to just give you 3 examples at the species, population, and individual levels. We're just starting out with a basic curiosity about nature. Today science is very much, it's always been very formal, but it's all about, going out and asking the right questions. There's a lot of emphasis on that. But a lot of our initial interest in science comes from curiosity and curiosity about nature. So I'm trying to kind of show how many of these explorations started that way. They led to, more formal research and insights, and how that eventually led to conservation, whether through management or policy or action and so on. So basically, the first story is about birds in the Western Ghats. And this is Robin, who used to be a postdoc in the lab. And he just walked into the lab one day saying that, I work on these birds. And when I worked on them on either side of the Palghat Gap, this is a deep valley in the Western Ghats, they don't seem to recognize each other's song. So maybe, there's something about this Palghat Gap. Maybe the birds across these 2 valleys of this valley are different from each other. So like I said, this basic curiosity. And then when we basically said, okay, let's look at all montane species, that is species which live at slightly higher elevations. The Western Ghats is not a very high mountain range, not like the Himalayas. So, but, above about 1,200 meters elevation or so, you have different habitats called the sholas, which are mixtures of grassland and forest habitats. And so we actually looked at these sky islands, or basically habitat islands, right? So if you're just looking at habitat above 1,200 meters, it becomes an island in the sky. And so we looked at these 4 habitat patches or sky islands for the entire montane bird community, all birds which live in these habitats. For example, this one over here is the very rare Nilgiri pipit, which we showed, through these surveys. So we started out by looking for these species in these areas. We showed, for example, that the Nilgiri pipit is only found in this, in B and C, and it only exists in, marshy areas above 1,900 meters. So its habitat is really, really restricted. So, but when we did this, when we looked at all of these different species, we basically found a really beautiful biogeographic pattern that, the shape of the mountain basically drives patterns of genetic differentiation and speciation for these bird species, even for birds which are, they fly. So you would imagine that mountains or valleys are not barriers, but they become habitat barriers. So even with change in climate, these montane habitats have been pushed up and down a bit, but some of the valleys are so deep that they were never contiguous. So they were pushed up and down the mountain, these habitats, but always the birds were disconnected. So in fact, 2 of these groups, they were called shortwings, but we renamed them Shola Kili, because that is the Kili is bird. And local people always call these birds of the Shola Kili. and the other, the laughing thrush, was renamed Montesincla. But basically, these were differentiated into, in the case of Sholakili, 3 species and a new genus, and in the case of Montesincla, 4 species and also a new genus. This was the common name, again, we used here is Chillappan, again, a name used by locals for these laughing thrushes. And this is just illustrations, but you can see they look kind of different. So initially we had only genetic data, which differentiated, for example, across the Palghat Gap, 9 million years, up to 9 million years, or 5 million years. So really deep divisions for mammals and birds across this, gap in the mountain. It's really a small gap. It's about 40 kilometers, the Palghat Gap. But it seems like it really is a biogeographic divide. Subsequent studies for frogs and monkeys and so on.
May I ask a question?
Yeah, please.
When you said that phylogenetically, you have evidence that this speciation is 9 million years old.
Yeah.
What are the error bars on those time scales of 9 million years?
Yeah, so maybe 1 million years or so, something like that. And basically, for the clades.
Okay.
Yeah.
After you finish.
Go ahead.
I'm going to ask you, because you started off by asking questions about behavior and song differences.
Yeah.
So are you going to correlate that?
So basically so the initial study was just to say, when we described it as a biogeographic divide, we saw that the valleys had similar effects on all species. That's what makes it a biogeographic divide, and not just something which works for this species or that species. Now, to define these as new genera and species, we used an integrative taxonomic approach, which we were not familiar with at all because we are not taxonomists, but we basically worked with data on song and morphology and genetic data, which allows you to basically say these are reciprocally monophyletic clades. That is, under no condition do birds mix with each other in terms of their clades. So based on these 3 lines of evidence, song, morphology, differentiation in song, differentiation in morphology, and differentiation in terms of the genetics, we were able to convince taxonomists that these are indeed, a new genus. A new genus also because they were called shortwing, and there are shortwings in the Himalayas, but they are not shortwings at all. So what we find with many species in the Western Ghats, they have long branches, so they seem to be quite unique. And if you look at India a little bit, you'll realize that the Western Ghats is actually a wet island in a sea of aridity. So most of India is very dry and the Western Ghats is wet. So almost like you could imagine islands in the sea, here you have these habitat wet islands in a sea of otherwise arid habitat. So basically the point was also that after we basically characterize these populations, and of course species are hypotheses. The more data we have more song data now. Robin has made a whole career on this, so he has song data now from thousands of birds and now genomes from hundreds of birds. So I'm sure there may be, there'll be much more to come on this story. But what happened was this resulted in highlighting this habitat, highlighting sky islands as very unique habitats which have endemic species which are old. The antiquity of these ecosystems was kind of highlighted. And in the recent revision of the Wildlife Protection Act, basically the Wildlife Protection Act is like the Endangered Species Act in the US. It's in India, the act which Indira Gandhi pushed in 1972 to conserve species. It actually does not allow hunting of anything. So it's a very progressive law. When this Wildlife Protection Act was revised, given this new information, so basically imagine earlier, since it was not known that Montesincla was 4 species, the species range was assumed to be all of this area, right? Now you have 4 species. So immediately the range becomes restricted. And once you have that restricted range, the endangerment status goes up. So basically following this work, which was actually very recent, right? 2017. So it's quite recent. But in 2022, when the Wildlife Protection Act was revised, all of these species were listed in Schedule 1. That means they have legally as much protection as tigers. Okay. So I think that's like, it was like something we didn't expect, right? But the fact that, there was this work which happened and there's a lot of bird watchers and people were really excited about these habitats, about these new species, led to kind of this movement of putting them on the Schedule 1 of the Wildlife Protection Act. So in some sense, that's research which directly has gone into policy, right? Without us just exploring this, they would probably just still not be on the act. Now, whether that helps the species or not is a different question. And maybe, unfortunately, the Wildlife Protection Act in India is more of a punitive act. It doesn't say, unlike say US Fish and Wildlife, where scientists are involved in planning, conservation, and recovery of species, the Wildlife Protection Act simply affords legal protection. So hopefully, though, at some point there'll be a change where there's more participation, a positive, like a plan for conservation as well. But it certainly highlighted these habitats and these species. Does anyone else have any questions? Go ahead. Yeah.
Just the sky islands were there long before the birds.
Yes, and in fact, there's very few studies on plants, but it would be really interesting to do ecosystem-wide studies where we look some species which are really old, like centipedes and stuff. They show more Gondwanan patterns, and they don't show. Basically, they show more. They don't necessarily show this relatively recent island type structure. Definitely, there are the frogs, tree frogs show it, bush frogs and tree frogs, and several mammals show it. We've just recently finished some work on elephants, which are so mobile, and they also show kind of these same, population structure patterns. But yes, the Sky Islands are much older. But sometimes with conservation, it's species which are charismatic, which tend to bring up habitats and conserve them. Okay, so the next kind of story is about tigers, and this is again something which was motivated by the curiosity of this student, Aditya Joshi, who was a master student in wildlife biology and conservation, and he was from Central India. He was from Nagpur, and he was really curious. So this is basically these black are the tiger reserves in India. Tiger conservation in India has been marked as a conservation success. It is in the Guinness Book of World Records, the largest conservation government-funded conservation effort anywhere in the world. Thousands and thousands of camera traps laid out across the protected areas used to use mark recapture-based estimates of the numbers of tigers. Basically, you capture a tiger, and then you see how many times you capture the same tiger and use a probabilistic approach to estimate the number of tigers. But basically, while that was fine, there's a huge focus on numbers. You can see that each of these protected areas is really tiny. Okay. And what we know from decades of ecology research on butterflies and many, many species is that there's kind of a minimum viable population size. If you have less than 50 individuals or 100 individuals, the chances of extinction because of demographic stochasticity and environmental stochasticity and genetics are high. So if we are to maintain tigers into the future, we still have on an average, the median number of tigers in any one of these reserves is about 30 or 35 individuals. So unless they are connected to each other, the sustainability of tiger population in the future is not very high, right? So there was no one at that point. This was probably, yeah. So you can see the first paper was in 2013, around 2010, 2011. Talking about connectivity. They were only talking about how many tigers, how many more tigers do we have this year, that year, and so on. So Aditya Joshi's, kind of curiosity was, are tigers moving between protected areas? And he, of course, the way to do this is, the hardcore, macho ecology way, which is to use radio collars and track animals, and watch them move. But how many animals are you going to radio collar? Right? It's very difficult to get permissions for these things. And you'd ideally want to collar, for example, in Yellowstone, every wolf is radio collared. We would love to do that, but it's very difficult to do such invasive research in India.
Difficult because of regulation, difficult because of resources.
Yeah, both. basically you have to import all the collars, and then there's all kinds of, because if you use satellite collars, you have to get all kinds of other permissions because, I don't know, some, you know,
The defense.
right,
related thing.
right, yeah. So basically, he started what's called landscape genetics, and this work was followed up by a student, PhD student, Prachi Thatte. She is a really, really talented young woman, and I was very lucky to work with her. I'll talk a little bit more about her later. But what this approach does is really simplistic. It's correlational. What it says is, okay, imagine that you have, all of these protected areas. You have tigers in them. You have genetic data associated with these protected areas. You have landscapes, real landscapes between them. If you have, you're looking at whether there is flow of genes between, these different landscapes, and the genetic sharing is kind of the truth. And based on that truth of sharing or not, and given the land use types which are, distributed here, you're going to infer whether specific landscape provide resistance to movement or not. The approach is called circuitscape. It's based on electrical connectivity theory. The populations are nodes, and you have a resistance between these nodes, okay? So basically, she and Aditya Joshi sampled across the central Indian landscape, mainly in Madhya Pradesh, Maharashtra, and so on. And based on the genetic data they collected, they inferred this kind of a resistance surface. So what you can see immediately, so this is not a map. This is the resistance map based on genetic data, resistance to tiger movement map. And when you kind of parse that out, what it tells you is that, populations in general are connected. So you can see there's a lot of white. So there are individuals are able to move. But high traffic roads and high density human settlements like cities are barriers for movement. And for example, this particular road is called NH7, National Highway 7. We also did some simulations to show what might happen in the future to connectivity given specific types of land use change. And we showed that, the probability of extinction in this landscape increases by 50% when this NH7 becomes a barrier. So now this NH7 is a highway between, through Madhya Pradesh, Maharashtra, Madhya Pradesh, Nagpur to Delhi, basically. And it bifurcates Kanha and Pench, these two famous protected areas. Pench is where Rudyard Kipling wrote Jungle Book. Basically, it's set in Pench. And Kanha and Pench are these really dynamic, populations which are outputting individuals, and the individuals are moving a lot here and there. And so this was an important connection to maintain, and disruption of that connection would have impacts for the future. So
Can I ask a
sure.
slightly more technical question associated with how these resistance maps were created? Because you said that it's associated with genetic relatedness, but you also build in aspects of urban and so how do you weight all these different?
Basically, it's a multi-parameter optimization, and you just say, okay, you classify, okay, this cell has road, this cell has, so you have five categories: roads, city, human density, agriculture, forest, something like that. And you basically ascribe resistance values to all of those.
But that's a bit arbitrary.
It is. And then you optimize by the observed sharing.
Sharing of
genetic share. So basically
genetic sharing only of tigers, or do you look
only
at
tigers. So this is only tigers. This is resistance map for tigers. So subsequently, we've now actually worked on 9 different species. And it's very interesting that, of course, the scale, some of them do. So some things are consistent, like roads and high density human settlements have negative impacts on all species. However, some species like muntjacs are the small deer, they are more, they require forest connectivity more than say tigers. The tigers are big and they don't need, they can walk through agriculture, they can, so they are probably least impacted by certain kinds of things, right? So the scale matters and also the kind of feature matters. So for example, very interestingly, Indian wild dogs or whole, which are pack living animals, they seem to be very strongly impacted by even small linear structures like railway lines, which other animals can seemingly cross. I don't know whether it's because they're social and the whole pack has to cross or something like that. So these are not mechanistic models at all. We have no idea why animals move the way they do, how they move. We have nothing, we know nothing about behavior as such, but we are inferring the result of it.
And are there ways to build exclosures and enclosures to try and see whether that will change, or that's just an impossible task in such highly urban?
Yeah. So basically, for example, what has happened in, say, in places like the US and Canada, there's funneling,
Yeah.
right? So basically, you just have a fence, and then you open that fence at an overpass or an underpass, and the animals are just funneled, and that's how they cross. Now, that works when you have large areas, right? We also modeled, for example, what happens if you just fence off all the protected areas. Extinction probability is very, very high. Basically, this whole landscapes are sustained because of movement. Without movement, they're not going to be sustained. The populations are just, the sizes are just too small of these areas. So I think, but funny you say that, because very recently in Uttar Pradesh, in the Tarai, they have just fenced the parks.
Despite the fact that that's probably
it doesn't make any
very
sense
bad.
biologically. So I'm not sure what's going to happen in the future, but there is a big push towards that.
And as a result of this, have you built any underpasses?
Yeah, so that's what I'm going to basically come to, right? So basically, this was, that's Kanha, this side, that's Kanha, and this is Pench. And I don't know how many of you are familiar with Indian politics, but there's a politician called Nitin Gadkari. So he is from Nagpur, and he's pushing, along with many others, a lot of development of roads, which is very important. I mean, a lot of India is quite remote. So one of the big things was to expand NH7 from a 2-lane highway to a 6-lane highway. Okay. And initially, Aditya Joshi's master's thesis was used to put a stay on this expansion. And it managed to do that for 2 or 3 years. Then, of course, there was a big push back. And after that, what happened was they started construction, they started this expansion. And at that point, an ex-alumnus, basically of the master's program, Milind Parivakam, we have this really interesting tool in India called the public interest litigation, which means as an individual, you can just go to court and challenge something and say, oh, this is not right. So Milind Parivakam, along with help from many others, launched a public interest litigation. And, our work, our paper and many other people's papers were put in court as evidence. And then basically after that, the court actually ruled that disruption, so this underpass would disrupt a natural biological process, which is the movement of animals between Kanha and Pench.
So it's expansion of the highway.
Yeah, expansion of the highway. Because, for example, I said that's one of the reasons why we actually did these simulations, where we actually said, what happens if this highway becomes a barrier? And it was, they were able to actually quote some numbers, right? And somehow that also matters a lot in court. They were actually able to say, okay, this is going to increase extinction by something, right? So, so basically then, all of this kind of came to a boil, and there was a lot of media attention on this, so the role of media is also critical. I don't think we should discount the role of media. And basically, that these 2 highways are going to cut these corridors, and this NGT is the National Green Tribunal. They also got involved. And so basically, at the end of it, it went to the High Court, and the court ruled or required NHAI, which is the National Highway Authority of India, to build an underpass.
One.
Okay. Yes, only one underpass.
And the expansion continued.
The expansion continued. So this is just a drone shot. It's really, you can see such beautiful forest on either side. Basically it's an overpass for vehicles, underpass for animals. And that is basically the overpass. It is the longest overpass in Asia. 700 meters is very long. I mean, maybe it would have been better to have multiple small ones, but anyway, that's what the court decided to do. And that's one view of it. Okay, it doesn't look very pretty, but that's Milind with the underpass, and basically it's working. So these are camera trap pictures from a year ago. Mothers and cubs, males, herbivores. These are dhole, gaur, all just using the underpass. So it looks like pretty trashy. I mean,
Yeah, exactly. I
you
was
know,
say that one.
but like all Indians, Indian animals also adjust. So, so it's just, I don't know, that to me, that was just like, that was amazing, right? That, so, like a lot of times we think of things in India as so difficult, bureaucracy, nothing works. But I think it's pretty amazing that in a span of 5 years or so, knowledge is actually getting incorporated into, in this case, concrete, right? It's actually having an impact on an underpass.
What is the length of the highway? Like the 700 meters is the overpass.
Yeah,
and what is the length?
well, the corridor is a few, like tens of kilometers. And there are tigers in that corridor itself. Kanha Pench corridor has become quite famous. There's a lot of wildlife living there, and we'll talk a little bit about in the end about the other problems which are now occurring. So there's a final story about, this is about individuals, and this is about these really beautiful black tigers. So these black tigers, this is from a place in Odisha called Simlipal Tiger Reserve, and there used to be ethnic people, tribals who lived in Simlipal, and they always talked about these black tigers. But everybody made fun of them and said, oh, you must be just lying. How can there be black tigers? It doesn't make any sense. Until in I think it was 1983, that a black tiger actually attacked someone and in his defense, this teenage boy actually with a bow and arrow, shot at the black tiger and it died. But basically that was the first proof that these such animals do exist. And, basically this was very fascinating from a genetics perspective, because it's not a melanic phenotype. It is what we call pseudomelanism. It's a disruption of pattern. The pattern is disrupted. So tigers, you can see this guy in the back, they have some kind of striping pattern, generally wider stripes, and then these blown up stripes in the center. But like these guys have just, the stripes are all array. So this is work by PhD students who's just finished, is moving on to his postdoc Vinay. But again, we got lucky here. So, initially it was just curiosity. Okay, why do these guys look like this? And also, why are they only in one place? So any population geneticist, if it's genetic, you should see polymorphism. You should see these animals in different locations. So why are these black tigers in only one location? So again, we got lucky. So luckily for us, so it would be really difficult to work on a phenotype in the wild like this, right? But we got lucky that there was a litter in a zoo which had, some black individuals. We were able to get the entire pedigree, and like basically we found the causal mutation. Again, we were lucky because some other people who've been working on cats, Greg Barsh and others, had been studying this type of phenotypes. And it turns out that in cats and cheetahs, there are kind of similar phenotypes. And there's a specific gene called TACPEP, and mutations in TACPEP, non-functional versions of TACPEP, result in disruption of pattern, whether it is spots in the cheetahs or whether it is stripes in tigers or stripes in cats. Okay, so we got lucky. We got really lucky. It was a simple mutation, single point mutation. And that was fine.
How hard, or so you use quantitative trait locus to assess?
Yeah. So basically we used association analysis in the pedigree, but for that candidate gene. So clearly we did association.
Because you knew about it
Yes.
in the cat.
Yes.
Okay.
So because we knew TACPEP is a good candidate locus, we sequenced whole genomes for 3 generations of individuals, about I think 12 individuals, something like that, which had and who did not have the trait. And then basically based on quantitative association analysis, trait analysis, there was a 200 times higher probability that it was this particular mutation. It's also missense mutation, basically in a region which was very highly conserved evolutionarily. So evolutionary analysis shows this mutation is never present anywhere else.
And it's the same that's also responsible for
different
cheetah?
mutation, same gene.
Okay.
Yeah.
In
different,
the cheetah, it's different. In the cat, it's different.
Yes. In all of them, different mutations, but the same gene. So yeah, so basically that was fine. But then we actually were able to go back into the field. And this is a place where tigers are really rare. So this was, Vinay did this field work in 2018, and he was walking in Simlipal 25 kilometers a day for 2 months or so, collecting fecal samples of tigers. Not once did he see a tiger. So really rare, like you don't see these animals. It's not like you can actually, see them and figure out this phenotype, right? So he was actually able to go to the field, collect fecal samples, identify tigers, make sure the fecal samples from a tiger, identify individuals, and then basically genotype these individuals. So because these were fecal samples, the DNA is degraded, but since we know the mutation, we can design a PCR such that you're close to the mutation. So you don't need to sequence the whole gene. You can just look at a small bit and genotype whether that mutation is present or not. And when we did this, it was really amazing that, basically we only found 12 individuals. So it's obviously a very small population, but the frequency of the mutation was almost 60%. So that is inferred from basically heterozygous, homozygous wild type, heterozygous mutation and wild type, and homozygous mutant, right? So based on these numbers, the allele frequency of the mutation frequency is about 60%. That was in 2018. And recent camera trap data, we've actually collected samples in 2023, which is 1 generation later, but we've not finished working on them, but the camera trap data suggests that almost 50 to 60% of the tigers are black now, which means that the mutation frequency is probably 75 to 80%. So it has already gone up. So what's happening here? Why is there some selection, or what's happening? So basically, what we believe is that, and we were able to show this, again, there's a lot of more details in the paper, that the reason why the frequency is so high is because of drift, just chance, and isolation. This is a very small and isolated population. And when we took our landscape genetic, inferences, and we laid it back onto this larger landscape, what it seems like is Simlipal is just simply too far away. There are no, there's all of Chhattisgarh and Jharkhand, where there are no tigers at all. There are no populations. So when I said earlier that, a lot of these landscapes are maintained by recolonization. So there have to be many sources, and animals should be able to move from one source to the other, right? So when you have a population which is so far away and isolated, even if that population is recovering, that is, there's enough prey and so on for that population, it's not connected to any others, it is isolated.
The heterozygous version is also
Is
black?
No,
Okay.
it's normal.
So,
Yeah.
so only
It's a recessive trait.
So it's a recessive trait, only homozygous dominant.
Yes,
Okay.
yes.
And yet, it's now starting to push through.
Yeah, so basically, the predictions are, because genetic drift, so the prediction is that the chance of fixation is higher when the frequency is higher, right? So chance of fixation is proportional to the frequency. So given that the frequency is 60%, there is a high chance that without gene flow, without influx of individuals, this particular mutation will get fixed. Now, does this matter? Well, it may not, that specific mutation may not have any effect on fitness, but overall, we would imagine that there are only 12 individuals or 15 or 20 individuals even. They've all been through a founding event. They're all highly rated and, of course, inbred. So basically, after we published this paper, in that paper, towards the end of it, we talked about what would happen to the mutation. Will it get fixed? And we also talked about this concept called genetic rescue, which actually has been explored in the US in the case of the Florida Panthers. The Florida Panthers did show inbreeding. They had, I forget what is basically undescended testes and kinked tails, phenotypes associated with inbreeding, but it was a huge, it was a big case in conservation where, in fact, when I was doing my PhD, it was discussed a lot. There were a lot of papers written about it. And finally, the consensus was that to rescue this population, it's better to bring in individuals from somewhere else.
It's effectively a gene flow.
Yes, gene flow into that population. This is called genetic rescue. And cougars were brought from Texas, and in no time at all of these inbred traits disappeared. So genetic rescue So what happens in inbred small populations is the bad variation is concentrated and
Starts getting fixed.
shared. It's shared because everybody's related. And so the effects of inbreeding show up. Even if there isn't inbreeding by choice, it's not necessarily that these individuals are mating with relatives by choice. It's not a mating strategy, but there's just nobody else and everybody's related, right? So in that paper, we suggested the idea of genetic rescue to not to offset bad mutation and also bring in potentially adaptive novel mutation. So this was, of course, this is in some sense, you're talking to managers who don't even know, who don't know anything about genetics, right? But very, very surprisingly, this year, after discussion, and this story, so this is crazy. This thing is, the black tigers, they look very pretty. So it was crazy. The media went crazy about this story. And everywhere it was trending, these black tigers and this mutation and spelling mistakes. And everybody was talking about mutations and DNA all of a sudden. It's so funny how some things people like or whatever, because they were able to see it, I think. Otherwise, you just say something, they're not, people don't internalize it. And for the first time now, the National Tiger Conservation Authority has approved the translocation of tigers for genetic replenishment.
So can I ask a question in that context? You said that tiger conservation in India really is taken off.
Yeah.
If you look at relatedness across these 30 or 40 different islands,
yeah,
what is known?
Yeah. So basically, that is, it's very beautiful. It's very geographic. So basically, Central India has the highest amount of genetic variation. And that is probably because it's at the center and it's a two-dimensional set of populations. Many others, like if you look at the Tarai in the North Indian foothills of the Himalayas, it's one-dimensional. Western Ghats, one-dimensional linear array. And overall, the numbers of tigers have always been very low in habitats like the Northeast, in rainforest type habitats, because they don't have that high prey density. So most of actually Central Indian stuff is quasi savanna, which has much higher predator densities. And so prey densities, which allows for higher predator densities. So basically Central India has the most amount of variation and other places have less variation. Now, so basically, it's funny because we try to push this whole idea of genetic rescue. And finally, this NTCA is the one which counts the tigers and all that. It's approved through an act of parliament. They said, okay, fine. There's permission now to move tigers. Now the question that came back, exactly, from where? And this is something which actually a lot of people haven't talked about. And if you look at the literature itself, there's a lot of hand waving that, okay, maybe slightly different, but not too different and all these things. But when we're talking about, we are able to now sequence genomes and we can talk about what's called genetic load or potentially missense mutations or loss of function mutations. Why can't we have a better strategy to suggest best possible sources? So we're actually kind of working on the go in some sense on saying, let's think about the source in terms of two axes. Evolutionary potential, quote unquote, which just means genetic variation. Some individuals have more genetic variation, or some populations have just more heterozygosity, more genetic variants than others. And that's good. We expect genetic variation in general is good. And also genetic compatibility. So if you have a lot of shared genetic load, that is shared bad mutations, that's not a good source.
So you would not know a priori where the shared bad mutations are unless you actually are able to connect it with a particular phenotype, right?
Well, these are putative bad mutations. Yeah. So that is still definitely in genetics itself, a big challenge right now, which everybody's working with, trying to understand how much do any of these affect phenotype. But a conservative assumption would be that, say, loss of function mutations, which basically render the gene to not have any function at all, can have an impact.
That would require lots of population level data. Do you have that?
No. So loss of function would be inference based on bioinformatics, right? It's a stop
Yeah.
codon or something like that. But yes, there are other approaches people are using. For example, you can look at whether specific genes are known to have mutations which have functional effects. So there's clinical databases, for example, with mice.
So this is all using homology of some sort.
Yeah, so basically you know that, so if you have you know that there's some mutation which is going to render this
Yeah.
gene like defunct.
In the mouse, and then you got to look for a homologous.
No, you have your tiger genome and you find that there's a mutation in some gene which renders it, that gene will not be functional. Now you look up these databases and see, are there clinical phenotypes in mice or other species where this gene is important? So if you find that there are phenotypes where this gene is important, you can sort of say that maybe this is potentially relevant functionally. On the other hand, there's a lot of human population level data you're talking about. There's something called a PLE score, where basically mutations present at very, 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 very low frequencies are probably, obviously they have some bad effect. Otherwise they should be at higher frequency. So you can use some, so that's something at the science end, we are working on that concomitantly, right? And also, I mean, hoping to characterize some of these mutations functionally in cell lines, but that is too slow. So the point is that we can try to push the science, but on the other hand, I mean, they need to move tigers in the next 3 months. So what do? Do you say, okay, let's throw all the science out and just randomly pick? Or this is what we understand so far. Let's do this. And then it's an experiment and we see what happens. So unfortunately, sometimes that is where we are at. Science is too slow for management and conservation. But this is something which basically then you can also do, you can do forward time simulations where you say, okay, and what should be the schedule be? Like, should you have one female every year, or, you know, how many years should you do it? Those kinds of things. So that's what we are hoping to do. And what's really surprising in this case is that the Odisha Forest Department is basically funding us. Now, it's not about the money, but it's basically that means they are willing to, they believe in it. They want to feel, they want to own it. And that's partly, I think that's part of the process, that if they participate like that, it's also their work. And then they are, it's not just some paper which we published, which they can ignore. Yeah. It's their work, which they have also been a part of. So anyway, so this is basically kind of the most recent example and most, I don't know, maybe we'll be wrong in what we propose, but it's also part of working with them and learning, you know, whether we are wrong or not. We'll see what happens. Yeah.
Professor Ramakrishnan, in humans, inbreeding consanguinity is related to ROH. Say,
yes. Yeah.
Segments of ROH.
Yes, ROH. Yeah.
Wouldn't it be an easier way of looking at it rather than looking at specific mutations? Because
sure. Yeah. So
although there are specific mutations, it will be masked by this expression. So I was
sure. Yeah. So we have looked at ROH. So this runs of homozygosity, and basically tigers have about 40% of their genomes in ROH, which correlates with the inbreeding coefficient F.
Yeah.
And basically what happens, what we think is that because these runs of homozygosity are homozygous, the deleterious mutations are enriched in these runs of homozygosity. So another strategy would be to break these runs of homozygosity by bringing in individuals who have contradictory runs of homozygosity.
That is fairly used in, for example, in say mice breeding. So
yeah.
Particularly at Jackson Labs,
right?
That is their bread and butter.
Yes. Yeah. So we are looking at it that way also. But basically, this load is another way to to also characterize how much of, I mean, mutation load is a kind of a well-known currency of population geneticists. But
1950 overloaded mutations by Muller.
Yeah. But basically, a lot of people are talking about load, and some recent papers, for example, are looking at mammoths and how load has changed over time and so on and so forth. But I think it's just one other way to look at what might be high load or low load. But definitely, if you have the same load, if it is potentially bad, you're going to have consequence of inbreeding, right? So something else I wanted to say. I'm not remembering. But anyway, so for example, sorry, I was going to say that with Asian lions, we've recently been sequencing genomes, and they have something like 70% of their genome in terms of homozygosity. So they're so homozygous, right? So a lot of times people say, why are you working on these species? They're so difficult and untractable. But actually, many of them are like inbred lines in the lab. So I personally also think we can actually learn some genetics from them. It's not just about, trying to understand them from a perspective of, and if we have been proposing model organisms for so long, saying that they are generalizable, then we should be able to look at these non-model systems and kind of first stuff. So I'm actually almost done. So yeah, so just thank you guys all for listening. And I'm really lucky to work with amazing people. I don't know whether you've ever seen this. These are elephants made from lantana. Lantana is an invasive species. These are life-size elephants. Actually, this guy who used to work with me called Tarsh, he and his wife is a designer. They basically started this, and these elephants have gone all over the world. They've made lots and lots of money. They've been on exhibit in London. Yeah, so it's really, India is a really amazing ecosystem, not in terms of the biological ecosystem, but it's really amazing, a lot of amazing energy, creative people. And I think that we shouldn't be too down about it. The pathways to doing things are not standard. There are no recipes or books. You have to forge your own path, but in a way that's kind of freeing. And I think that you can actually do a lot if you do that. So I'll just leave you guys with some nice pictures. So this is a really nice photo montage of a tigress outside a protected area. This is outside Bandhavgarh. It's a camera trap image. You can see this is at 9:15 p.m. and there she has 3 cubs. This is at 11:30 p.m. And this is the same spot at 7:40 a.m. and 12:45 p.m. So really, people and these large carnivores, the largest carnivores on earth, are really sharing space, and that it can continue and wildlife can thrive and survive is quite exceptional. That's it. Thanks. Stop there. Happy to take any further questions.
I think it may be best if we just open it up immediately to questions and comments.
Yeah.
Professor Ramakrishnan, you mentioned that it's a very fascinating one on birds.
Yeah.
This is almost like island biogeography.
Right.
And approximately a similar one was, of course, described by Darwin. And then later, Grant also published the quantitative genetic analysis for having worked with Russ Landing.
Right.
Have you done any quantitative genetic analysis on that? Those?
No. But it would be there. So the problem is that, I think we need a lot more basic genetic data to start doing those things. A lot of genomes, a lot of pedigrees and stuff like that, which we don't have so much of. Long-term studies like the Grants did or some others have done are very, very rare globally, actually. And so these long-term studies, I think it would be good to start some of them in India so that we can look at these kind of things. Also things like song, changes in song. So Robin, for example, has done some work looking at variation in song across the Western Ghats. And there's some evidence of cultural transmission, as well as what seems to be a mirroring of biogeographic patterns and so on. So I think there's a lot more work to be done, but unfortunately the community is very small. So I don't know how much of it will happen.
My second question is in our title, completely armchair biologist here. So this is one thing that I always wanted to do since 1980, which I was saying. But on the other hand, the tiger population, what I could feel is almost like the central population, island populations, and then when you do, either using molecular system or quantitative, just morphological morphological data, do you see any something like geographic, so genetic distances? Are they related or they unrelated?
Yeah. So
or
basically,
just random.
you see geographic isolation by distance. You do see isolation by distance, but isolation by distance breaks down at around 50 to 100 kilometers, which is roughly like dispersal distance, very roughly speaking.
I thought for things like tigers, it should be much
It should
more.
be higher.
Yeah.
So 200 or so it should be. But I think basically India used to be packed with tigers, right? Like there were tigers everywhere. And so then the effective dispersal distance may not have been very high. Now I think dispersal distances are higher. There are records of tigers moving 1,000 kilometers and all that stuff. So I think this overall, they probably did not realize the possible dispersal distances until relatively recently. But yes, you do have isolation by distance and all that. But I think there's been too much of gene flow, too much of mixing to see any very strong clinal variation. People say, oh, the tigers here are oranger, the tigers there are whiter, but I don't know of any good study which has investigated that. There are some studies which suggest that tigers in the Sundarbans have smaller skulls, smaller in general, because they also tend to eat, supposedly they eat fish and all that, which is much smaller. In general, carnivores eat about 1.2 times their body size, so larger things. But there's no such well-studied pattern documented.
Have you, along the same lines, has any one of your students worked on population differentiation, FST, and other things?
Yeah.
So, what is the range of FST?
Yeah. So FST is about 2.25 to 0.3. So not very high, but not very low. Between tiger populations.
Can I ask whether there's so many tigers in captivity?
Right.
And is there any, is it useful at all to think about this could be a gene pool that could help?
Yeah. So basically, so this paper I was just telling you about, where I just helped a little bit, but this team from Stanford, they collected something like 400 individuals from across the US in captivity. And what they have found is most of these are in rescue centers and so on. And all of those individuals are of mixed ancestry. So at some point, because of this fascination for white tigers, some individuals were brought from India. So the original white tiger is a guy called Mohan from Rewa. And, and all of the white tigers are his descendants. But basically that was the one Indian ancestry. And then most of the others, so all of them are quite admixed. So I think one of the potential useful aspects is in studies of things like inbreeding. So for example, phenotypic studies with zoo animals are much easier than in the wild. So that's something we are trying to do, for example, but they have variation, but what I'm not sure unless we understand what is good, adaptive or not, what can we actually, how can they actually be used for conservation?
I'm thinking as far as like, could you introduce one of these individuals into that population of black tigers, or could it be?
Yeah, you could. So basically, for example, now there's there's a big push to in Madhya Pradesh to introduce white tigers, because people say, well, the white tigers were driven to extinction by humans. They were so prized, people shot them. So why not we introduce white tigers? Now that is motivated more by finance, so tourism and revenue from tourism. But if you think about it, white tigers today are only in zoos, and most of them are very highly inbred. So you're introducing white tigers, but you're also introducing a whole lot of load. So it's unlikely that most individuals in zoos, however much value plan mating, they still are relatively inbred. But in India, there is no planning in zoos at all. So there's a lot of mating between relatives. So in a way, I'm trying to work with that, because they don't even have a stud book. So we're first of all starting to build a stud book for them, and they have, SIB matings and so on. So we can actually use those to look at inbred traits and inbreeding associated traits. Yeah.
More questions.
Next one,
Yeah.
what is the, on an average, what is this, in your estimate, is effective population size in tigers? Any range?
Yeah. So basically effective population size is around, it's low compared to humans. It's about 1,000 to 2,000. Yeah. But basically the signatures we're seeing in India and all, or overall globally, there's a lot of founding events, right? So basically the other thing which we have just started doing is to work with trophies. So basically between maybe like 1850 and 1972 was when most like tigers were shot like crazy, right?
What was the population, if you have a sense, at, for example, in 1850?
They were probably, at least even in India, probably
100,000?
hundreds of thousands. Yeah, hundreds. That's the guess. But this is based on demographic reconstruction. So,
It's a lot of error bars.
yeah. I don't know how much we can trust that. But so basically, what we're trying to do is to look at, we've been sampling these trophies. So for example, Chhattisgarh, there are no tigers, but there are a lot of tiger trophies in Chhattisgarh. And we are sampling those from whenever they were shot. And we've just set up a lab to work with archival DNA. So we're getting this DNA, and then we can actually also look at things like the load, mutation load. If there was something which was a lethal or something like that, it should be completely going to a frequency of 0. So you can statistically infer these things, but which mutations those were, which had consequences harder. So maybe with these kind of data, we can also look at what happens is when you have very strong bottlenecks, effective size becomes hard to discern because it is much more affected by the bottleneck because there's a harmonic mean. You can't tell what it was in the past. So it will be really nice to reconstruct these, basically look at both areas where tigers are extinct, like Chhattisgarh, Afghanistan, Pakistan, for example, there were tigers all the way over there. And also through time, how genetic variation has changed to understand more about the trajectories of these populations.
Maybe you should go to all these mathas, the Swamijis used to sit on them.
Yeah, so the problem is the Swamijis, they get donations from people. Like a lot of people give them, so they don't, so because we need to know geographic and time stamped data.
Is there any possibility, or have you already done analysis of the tigers in other parts of Asia? So in Indonesia, Malaysia,
Yeah.
and then China, and Siberian tigers?
Yeah.
Very completely different.
Yeah. So we did some work on that range-wide variation, and basically, overall the divergence times are very recent, like 10 to 20,000 years, and probably because they were so contiguous, right? Very interestingly, you see, for example, tigers in Northeast India have some admixture from Southeast Asia and so on. So you do see this relatively contiguous genetic variation. But tigers in India as a whole have much higher genetic variation, 70% or more of the species genetic variation compared to, say, in the Russian Far East, very low genetic variation. So in that sense, Indian tigers are kind of the repository. That's where a lot of the genetic variation is, because they are also very abundant and in different habitats.
Could you talk a little more about how the variation at height of like valleys and mountains have an impact?
Yeah, okay. Can I use the board? Sorry, I don't want to take a class, but so basically, we did a cross section of the mountain. It goes like this. So basically, if you're living at the tops, right, the habitat with climate change may be pushed up and down a bit, but the montane habitat will never go all the way down. So when the valley is very deep and wide, it will always be a barrier for these high elevation habitats. And anything which lives at high elevation will respond the same way, because if it's tied to this habitat, right, then this is always a barrier. And with all we know about evolution biology, allopatry, separation or the lack of gene flow, is what results in differentiation. So over time, basically, these have become barriers. Does that make sense?
Yeah.
And the shallower the valley, the lower it is a barrier, because suppose, this valley, for example, if the habitat is all the way down here, there's probably gene flow somehow or the other. But this is just 2D. So the shape of the mountain basically drives diversification. It's a nice thing to think about it like that.
More questions? Krishna, thank you so very
Thank you
much
so much.
for the
Yeah.
time and for a wonderful talk.
Thank you. Thanks, Dr.